What if a tiny glow can tell us something really important about what is happening inside of a small molecule? Hello everybody, I'm Molly and I will be taking you through the sensing genetic disorders with fluorescence activity. In this experiment, we will learn how different compounds fluoresce and how this is being used in research. The background for this can be found in this video as created by the second year chemistry undergraduate students. It explains how fluorescent sensors can be used on DNA. The aim of this experiment is to make a selection of fluorescent solutions and a molecular fluorescent pH sensor. This is what you will need. So first you're going to need a UV lamp. You are also going to need a range of beakers, conical flasks, and stirring rods, some filter paper and filter funnels, some spatulas, ethanol, hydrochloric acid, and sodium hydroxide, spinach leaves, non-bio washing powder, turmeric, tonic water, vitamin B2, which is also known as riboflavin. I would recommend getting these as capsules, distilled water, which I don't have shown here, and universal indicator pH paper. This is how you complete the activity. Teachers, if you are using this as a part of your lesson, you may want to show this either before or after the students have a go themselves, depending on what works best for your class. So firstly, we're going to make up 5 different fluorescent solutions from materials that can be found within the home. In order to make the ethanolic chlorophyll solution, crush up 5 grams of spinach leaves in 25 milliliters of ethanol. So I'm going to put my 5 grams of spinach into this plastic bag, and then I'm going to measure out 25 milliliters of ethanol, and I'm going to pour this into the bag, let out all the excess air, give it a little thingy, and then crush it as so. You may have to do this for several minutes. So once your solution has turned quite a dark green, we're then going to filter this. So what I'm going to do is I'm going to get my filter funnel and filter paper, get a single sheet of the filter paper, fold it in half, and half again. And then open it up, like so. Very importantly, I am filtering this into a pre-labeled tube, so I know exactly what it is. I'm going to keep this filtering while preparing the next thing. Next, we're going to make the ethanoic humorin solution, where what we're going to do is we're going to dissolve a spatula's full of turmeric in 25 milliliters of ethanol. So first, I'm going to once again measure out 25 milliliters of ethanol. And it says a spatula full, which is about 0.3 grams. And I'm going to mix until all of this is dissolved. So once most of your turmeric has dissolved into your ethanol, what you can do is then filter it as well. Okay, next we're going to make the aqueous still bean solution. I'm going to dissolve 1 gram of the non-bio washing powder into 25 milliliters of distilled water. Try and stir this carefully so that it doesn't create loads of bubbles. And then we're going to filter this and collect the colourless filtrate into, once again, a labelled container. I'm going to let that filter and carry on. So for the aqueous quinine solution, all we're going to do is pour 25 milliliters of tonic into the flask. And finally, we're going to make up our riboflavin solution. What you need to do is break open the contents of one of the tablets into 50 ml of distilled water. So if you grab both of the ends and twist, the capsules seem to come open. And then what we got to do is we got to stir this thoroughly. Note that most of this won't dissolve, but that's okay. Just give it a really good stir. And then once again, I'm going to filter this. I'm not going to pour all of this into my filter because my flask only contains 30 milliliters, not 50. And I'm just going to let these all filter, and I'll cut back to you guys when I'm done. Okay, so once these have finished filtering, and I've already done this for the ethylenoic coumarin, all you can do is take the funnels out, bin the filter paper, and wash your funnels out. Okay, so now that you have all 5 of your solutions ready, you can then test these under the UV light. Okay, so to test the fluorescence of the different solutions we've just created, I've set up this system here, which is quite simply just a cardboard box covered with a lab coat. I've put my camera in the lab coat so that we can see via video the fluorescence. However, if you're doing this in the lab or at home, just pop your head under the lab coat just up here and you should see it clearly. So first, what we're going to do is we're going to see the different solutions in normal light conditions, also known as the torch end of this thing just up here. So any just white light torch will do for this part. And as you can see here, this is what they just look like in white light. Very similar to when we were creating them and purifying them. However, if I switch to the UV section just here and pop it in the box like this, you can see them all fluoresce. If I take the UV light away, it disappears, and if I bring the UV light back, you can see it. Okay, so I've put the chlorophyll in next to the UV lamp just by itself so that you can really see how the fluorescence of the chlorophyll is more on the red end of the spectrum, as I was trying to show earlier. So my camera picks this up better on its own because it can see the darker light on its own easier. To make our fluorescent sensor, what we're going to do is we're going to add 2 lots of 5 milliliters of water into here, and then we're going to add 1 milliliter of our solution to each of these. This bit here is 2.5, so we're going to go up to that line. And then I'm going to test the pH of these with some universal indicator strips. Yeah, there you go. And as you can see, it looks very similar to sort of pH 6, pH 7. Now we're going to get our hydrochloric acid solution, and in one of them, just one of them, just add a few drops of this until the pH goes down to 1. And test the pH again. So this is more like pH 2-3, so I'm just going to add a couple more drops. Nice and red. So I know that the pH of this now is either 1 or very close to 1. So now that we know this one's pH 1 and this one's pH 7, we're going to look at under the UV lamp again. So I'm going to put some lids on so that I don't accidentally spill this. And then once again, as you can see, with the white light of the torch, they look very similar, but with the UV light, they look quite significantly different. So you can see just up here that the pH 7 is a lot more fluorescent than the pH 1. I'm going to actually add a couple more drops of hydrochloric acid to the pH 1 just to see if I can really see even more of a difference. Once again, with the white light torch, very similar. And the UV torch, yeah, look at that. So as you can see, the lower the pH, the bigger the difference you can see. Okay, so now the really cool thing about this is that you can reverse this effect. I'm going to take the pH 1 back out, and I'm going to add a few drops of sodium hydroxide to turn this back to pH 7, and we're going to see what happens. pH 7. So once again, put the cap back on this, and we're going to take you back over. Once again, as always, first checking with the white light. They look very similar. So the one on the left-hand side is the original pH 7 that I've not touched with any acid or bases. The one on the right-hand side, I've added acid to it, took it down to pH 1, and we saw that the fluorescence turned off. And now I've tried to re-neutralize it with a base to turn it back to pH 7. As you can see here, the fluorescence has come back. Purely from my curiosity, I'm going to add some more base back on the one on the right hand side because I want to see if it has a difference. This is now really high pH. Go with a normal white light torch as normal. As you can see, this is pretty standard. Let's go in with the UV light. And as you can see, it's only at pH 7 that it fluoresces. Now, you can try a bunch of different pHs on the pH scale by adding them single dropwise. There are a set of questions in the activity pack on the ChemBAM webpage for you to check out at this point. Let's go into the science of how this works. Please feel free to use this as part of your lesson. It's easy to see with your eyes that fluorescence is different at pH 7 compared to the riboflavin solution at pH 14 compared to pH 1. But it's still not easy to see the difference between these two solutions. It's also possible to analyze this more accurately using a technique called photoluminescence spectroscopy. In the resource pack, there is a bunch of data that you can use to plot these graphs yourself. In the experiment we just completed, fluorescence molecular sensors are being used to detect changes in pH. In the School of Chemistry at the University of Birmingham, researchers working with Jim Tucker have been using a similar concept to detect base changes, also known as SNPs, in DNA. To be able to detect which SNP is present in each section of the DNA, the researchers first make a short strand of DNA that will match one form of the SNP. This has a fluorescent probe at one of the bases, which makes it a fluorescent sensor. When the fluorescent sensor is added to the DNA, it binds with DNA to form the double helix. The fluorescence of the sensor will be different depending on whether the DNA bases are matched or mismatched because the local environment around the fluorescent molecule changes. Therefore, the fluorescent output changes with the SNP input, and this can be used to detect various diseases. You can find more out about this research on our webpage. I hope you've enjoyed this fun activity. Please check out the other experiments, videos, and resources on our website. And if you have any questions, feedback, or would like to request a visit, please use the form in the Contact Us section to get in touch. There are also some other videos that ChemBAM has made, so if you are interested, please check out the YouTube channel. Thank you so much. Bye!